Welcome Genetic Teacher channel. Today, we are talking about bacterial spread plate culture method that initiated by Pipette small volume of diluted liquid bacterial culture over surface agar medium by rotate L-shape spreader over surface agar medium evenly for spreading by adding 0.1 milliliters diluted liquid bacterial sample over surface agar media by dipping L-shape spreader in 70% ethanol alcohol. Then, flame it, and allow to cool. After that, spread bacterial sample uniformly near flame, and incubate all culture plates, in inverted positions, as mentioned in next slide. Thanks for attention. Please like and share and subscribe Genetic Teacher videos and channel.